I'm the head of innovation at Swissnex in Boston, and it's my pleasure today to be the moderator for our event. It's also my privilege to introduce Benjamin Bullman, our CEO.
Thanks, Alicia. Hi, everyone. So my name is Benjamin. I'm the CEO of Swissnex in Boston and New York. Swissnex is a global network of science and technology hubs that is established by Switzerland abroad. We have locations in the US, in Brazil, in China, India, and Japan, as well as additional colleagues located within about 20 Swiss embassies all around the world. Our mission is to unlock the potential of education, research, and innovation to tackle the big challenges of our time. As a Nexus, we do that by connecting people, connecting ideas, connecting resources across geographies, of course, but also across sectors and across disciplines, and by turning those connections into concrete actions. And that's precisely what we're doing today. We have an amazing group of Swiss BioTech and MedTech startups here that are tackling pretty big challenges like cancer or aging. And so they are building bridges across Switzerland and the US, but also between biology and engineering. And from a Swissnex perspective and a Boston perspective, to be more precise, this interface between biology and engineering is poised to be the technology revolution of the 21st century. In that sense, today we are offering you a front row into the future, and we hope that you very much enjoy the ride. And that said, it's my pleasure to introduce you to our partner organization, VentureLab, and more specifically to its co-founder, Jordi Montserrat. He is behind this adventure and behind bringing all these startups to the East Coast today.
So it's a great pleasure to be back in Boston after 2 years of, as we know, a hold due to the COVID. So great to be here. I have the great pleasure to introduce VentureLab maybe in a couple of words. At VentureLab for over 15 years now, we've been building several platforms in order to support entrepreneurs and to give them a chance to grow and to expand internationally and support them in their fundraising. With this particularly, I'd like to mention for those who are interested, we are running VentureKick, which is a national program in Switzerland, which does pre-funding, pre-seed funding. We are running the top 100. All of those who are interested to see what those startups become on a national global base. We have the top 100 Swiss startups, which you can download. The recent ranking was published beginning of September. And of course, the VentureLeaders, which is this program over 15 years coming to Boston and bringing top-level startups, the one that you will have the pleasure to see today. And over this time, it's my great pleasure as well to thank the partners, because of course, without them, this adventure would not be possible, in particular here, Swissnex. Thank you, Alicia, and thank you to the whole team and to Ben for welcoming us. Back in Switzerland, our Swiss Institutes of Technology, which you know are holding top-level rankings on a global base, Hans-Jörg Charitable Foundation, Kelle Hals, the PSI, the University of Zurich, Canton and Vaud and Canton Zurich are the ones who allow us to make this, and here in Boston, Morrison and Foster. Without further ado, I think that we're going to go into the startups because that's the essence of today's event. Alicia, I leave the floor. Thank you.
Thank you, Jordi. Thank you for being with us. It's always a pleasure. So it's my honor to introduce you to our first startup. We have LifeMatrix Technologies. Robin will be coming up to join us. So LifeMatrix is creating biomimetic implants designed to grow and regenerate. All right, over to you.
All right, so behind me on this picture, you see George. George was born with the congenital heart defect, which means that he needs to undergo surgical intervention at a very early stage of his life. He will receive a synthetic implant, and now while George will grow over the next few years, the implant he received simply doesn't. My name's Robin, I'm co-founder at LifeMatrix, and we want to change that. We have developed a unique bioengineering technology which allows us to produce implants which after implantation transform into living tissue and have the capacity to grow and regenerate. Therefore, they offer a lifelong solution to George. What makes us very excited is that our technology allows us to produce a wide range of different applications. Therefore, we can not only serve the needs of George and other children with congenital heart defects, but all patients in need for long-lasting cardiovascular implants. We have successfully proven that for several applications such as vascular grafts, transcatheter heart valves, septal occluders, and cardiac patches. In order to approach the huge market potential of $16 billion for cardiovascular implants, we intend to identify the best partner for each of our indications and generate revenues through co-development and licensing deals. At LifeMatrix, we have successfully demonstrated clinical-grade long-term safety and efficacy in translational sheep models for various indications. Our first product, the LifeMatrix Vascular Graft, meets the compliance requirements of GMP and ISO 13485. We have established a regulatory pathway and have initiated the interactions with FDA in order to start a first-in-human clinical trial in the second half of next year. Our preclinical development is supported by V Zurich, a joint incubator by the university and ETH Zurich. We have a strong IP portfolio, which we have licensed from the university, and we have secured our freedom to operate with a supply and license agreement with one of our key suppliers. We are now looking to find investors to join our Series A round of 12 to 15 million, where we want to establish our standalone commercial manufacturing unit, complete the first in-human clinical trial of our first product, and to also continue the work on our product pipeline. If this has sparked your interest, I'm very happy if you reach out to me, and I'm now available for any questions you might have.
Great. Thank you so much for your presentation. We can take questions from the audience if anyone would like to type in a question, or you can ask it verbally. We have about 2 minutes for any questions. In the meanwhile, I think I can ask, so you've been in Boston for about a week now. Is there anything that you found surprising here that will help you with your business or maybe become a challenge?
So, of course, it is always a challenge to enter into a new country where basically your network is not as well established as before. But so far, it has been a very great experience with the help of you guys from Swissnex and, of course, also the peers we are within this group to make a first step into this next adventure for us, the US.
Wonderful. And if you could meet one type of expert in Boston your last remaining 3 days, who would it be?
So for us, definitely the regulatory aspects are always challenging as they are different in every country. And then, of course, we are mainly interested in meeting investors who might want to support us in our adventure.
Fantastic. Are there any questions from the audience? Oh, yes, we do have a question. So how does the graft vascularize?
So basically, in our production, we use 2 different components. So we have biodegradable polymer scaffolds, and we seed them with human donor cells. We then culture these scaffolds and the cells where they form extracellular matrix in the lab, and we remove all the cells of the final product. So our final product is a composition of biodegradable polymer and human extracellular matrix. And this composition is then actively supporting cell infiltration after implantation, where basically the originally implanted structure will be degraded and replaced by native-like healthy tissue.
Fantastic. Very, very novel technology. We're looking forward to having you partner in Boston and wishing you the best of luck.
Thank you very much, Alicia.
Thank you. All right. I think it's time now that we can welcome our next presenter to the stage. Please join me in welcoming Philip from Araris. They are building best-in-class technology for antibody-drug conjugates. Over to you.
Thank you very much, Alicia, for the kind introduction. Hello, once again, my name is Philip. I'm the CEO and co-founder of Araris. It's a big pleasure to introduce you here to Araris, and Araris is focused on delivering best-in-class technology for antibody-drug conjugates. So, I mean, the concept of an ADC or antibody-drug conjugates is quite an old concept. I mean, ADCs, they have been around for over 30 years. However, And when you look at how the concept works, it's basically like this, that you hook up a very potent payload onto an antibody, the process of which is called conjugation. Then you inject the ADC into the patient, where the ADC then selectively binds to the tumor cell, which then leads to the internalization of the ADC and degradation of the ADC, and therefore the payload will be released and then cell death. I mean, the concept is quite simple. I mean, you only need to attach a payload onto an antibody. However, it's quite actually challenging. And over the 30 years, ADCs have really suffered from a lot of setback, mainly because the drug is randomly attached in an unstable way. The payload is very hydrophobic. It's also a problem. And also the generation of an ADC itself is quite challenging. So when you look at the most recent ADCs that have been developed, and also the technologies, they still suffer from poor stability due to the design. And when you measure the concentration of the ADC, and also when you measure the concentration of the antibody, you see there's a clear difference in concentration. This is shown on the top left, indicating that the payload is lost. And when the payload is lost, actually you have will have very poor efficacy, and the tumor regrowth will rapidly regrow, and it's shown on the right. And also because the payload is lost, you will have tolerability issues, causing then limited therapeutic index. And also attaching the payload onto an antibody is quite challenging. This is shown on the bottom, because you need several steps to attach a payload in a very well-defined manner onto an antibody. And this is where we come in with Araris. We have developed a link technology that enables to address these issues. So on the top left, when we measure the concentration of the antibody and also the payload that is attached on it, where there is a perfect overlay, indicating that our link technology enables to generate super stable ADCs, which have very high efficacy and also an improved tolerability. And on the right, you see what this actually means. Thanks to our link technology, we can generate very stable and efficient ADCs that have a very high efficacy at very low dosing, because we can deliver a lot more toxic drugs to the tumor compared with any other link technology in the world. And also the manufacturing with our link technology is quite improved, because we can, in one step, we can attach a payload onto an antibody, actually 2 or 4 drugs, very rapidly. And this is quite actually completely unique in the whole ADC space. So as you can see, we address all those issues and can generate very stable ADCs that are able to deliver very efficiently high amounts of toxins to the tumors. And we've also data showing that. Last slide. So we are now in the process of raising a 55 million Series A, and the goal is, with our lead candidate focused on CD79B, to show a clinical proof of concept that we have superior efficacy and also tolerability compared to what's out on the market, which would then immediately validate our asset as well as our link technology based on that. And in addition, we're also having a new target, actually a best-in-class or first-in-class ADC, actually, that we would like to develop in IND-enabling stage. And also we have platform development. So with this, I'm at the end. I'm very happy to take your questions now. And we have also actually data showing what we claim here. So thank you very much.
Fantastic. Thank you so much for the strong presentation. Can you tell us a little bit more about the indications that you think are most valuable for this technology?
So the route we would like to go, we would like to focus on a mixed balance. So with the CD79B compound, we have a balanced, because the target is validated, the antibody we use is validated, as well as the payload is validated. And therefore, it's sort of a medium risk approach, while the second one is really high risk. It would be first in class or best in class ADC. So, and the indication for CD79B would be DLBCL. So, yeah.
And we have a question from the audience. Doug asked, Doug, who was here yesterday, hello Doug, asked if you are familiar with Mythic Therapeutics, which is a Boston area company also apparently developing ADCs. Are you familiar with them?
Yes, I'm familiar with them.
And can you compare your technology a little?
I think what Mythic has developed, I mean, from what I see, what has been published in terms of patents, I think they're rather focusing on like developing antibodies that selectively bind only under certain conditions. So they're rather focused on the antibody. We focus on the linker, which we think is quite important. And they're, yeah, exactly.
Fantastic. I think that might be all the questions we have time for. But if you're interested in getting in touch, please do. And thank you so much for your presentation.
Thank you very much, Alicia, too.
Ariadne is next generation intelligence for drug discovery in the neurological space. Welcome to the stage, and the floor is yours.
Thanks.
Hi everyone, this is Fabian Svara, co-founder and CTO at Ariadne.ai. At Ariadne.ai, our mission is to revolutionize drug discovery for neurological diseases by combining human and artificial intelligence. Let me start by introducing the team. We are a fully founder-led and founder-owned company at this point, combining more than 30 years of combined research experience between us, gained at some of the most prestigious research universities and institutions in the world focusing on neuroscience. Before I start with what we do, neurological diseases in general are a huge problem for humanity as a whole. At this very point in time, 1 in 6 humans suffers from some sort of neurological disease, meaning that more than 1 billion people are affected in this very moment. There are more than 1,000 known brain diseases, and more likely than not, one of them is going to affect us in our life as well. However, cures are terribly lacking, and one of the big reasons for this is that it is incredibly difficult to lead a successful drug discovery program for a neurological disease. The failure rate is as high as 99.6%, and one of the big reasons for that is that there are no good biomarkers that can tell you what the status of the brain is in the process of a neurological drug development program. And our solution to that issue is LMTrace. LMTrace is an imaging-based AI-powered biomarker. In other words, it's a technology that allows you to go from microscopy images to incredibly detailed analysis that can tell you a lot about the health status of that piece of tissue and what your intervention is doing to that piece of tissue. So in contrast to classical cell counting methods used previously that can give you maybe the density of cells or very coarsely whether a cell is dying or not, LMTrace gives you the precise three-dimensional shape of every neuron, including the fine arborizations that are incredibly difficult even for a human to trace out manually. And with a suitable microscopy method, we can even recover the synaptic contacts and thus the connectivity in that piece of tissue, allowing much more accurate target validation, lead testing, and drug discovery. We've proven traction with more than 50 clients on 3 continents so far. The total addressable market in neuroscience R&D is estimated to be 46.5 billion euros. And based on a few simple assumptions, we believe that our SOM is in the range of 500 to 630 million. Now, as of this year, we've generated more than 1.22 million euros just this year. We're a cash flow positive company, we're profitable, and as a sort of strategic mode, we've generated at this time more than 100,000 hours worth of manually labeled training data and more than 150 terabytes of diverse biomedical microscopy image data in our ground truth database. So in the future, our goal is to industrialize LMTrace to make neuron shape analysis into the new standard for neurological drug screening. To this end, we want to focus on scaling the technology to industrial throughput. Second, developing the business and commercializing. And third, developing an in-house microscopy assay pipeline to really prove the revolutionary potential in-house with known active compounds. With that, I thank you for your attention and I'm happy to take questions.
Great, thank you for your presentation. Are there any questions from the audience before we launch in? I think we have time for about one question. Your question is going to be, so you're coming to Boston for a specific type of partner. Could you talk about who would be the best people for you to talk to over the next 3 days?
Yeah, the best people for us to talk to over the next 3 days would be people who share a vision that structure and determines function in the brain, and that this is a biomarker that deserves growth funding at this time to really accelerate our growth trajectory beyond what we've been able to achieve organically so far.
There are 2 questions that we would like to get to in the audience, if that's okay, Alicia.
Yeah, please.
One is, what is the nature of the revenue? And the other is, what is the specific disease state you are targeting?
Sorry, I didn't hear the
first one. What is the nature of revenue?
Okay, the revenue is generated by clients who approach us with a problem that they're facing where they're generating too much microscopy data essentially to handle in-house, and where they need someone to develop a pipeline that automates that for them and generates basically analyses that are at the level that a human would otherwise need to perform manually. And
so services revenue, basically.
And are there any specific disease states that are preferred?
Yeah, we're focusing on preclinical research. So we're working with model organisms, with model tissue, and focusing essentially on ex vitro samples.
Great. Thank you so much.
Thanks.
It's my pleasure to introduce Elena of KetoSwiss. They are mitigating metabolic migraine. And I know Elena has a very personal story with this. We're looking forward to hearing your presentation. The floor is yours.
Thank you, Alicia. Hello, everyone. I'm Dr. Elena, a neuroscientist, PhD in clinical research, and the founder and CEO of KetoSwiss. I'm basically here today to tell you why this is no longer me. These are my attempts to illustrate what it's like to have chronic migraine about 7 years ago. Migraines are actually the most common neurological disease and the third most common of all diseases worldwide. It's extremely expensive for society, and apart from these recent injection-based chronic painkiller drugs, there's no migraine-specific prophylactic agents out there. What if I told you that migraines, or at least a subgroup of migraine, are actually as much a metabolic disease as they are a neurological one? Despite food having never been as abundant as it is today, what we eat has changed drastically. And very simply put, on these modern Western diets, migraine brains are starving. This energy deficit in the brain, or the increase in oxidative stress in the brain, could be at the very root cause of migraine. But why is that a good thing? Well, we cannot do much about our neurological hardwiring to date, but we can fix metabolism. And this is what we do with MigraKete, our metabolic migraine prevention product. The concept behind MigraKete has been awarded several prizes, was recently published in Nature Neurology, and is also patent-backed, with the first of 4 patent applications having all claims approved in the US already. It's also safe and seems to be efficacious in this newly defined metabolic migraine subgroup. In a recent phase 2A randomized controlled crossover efficacy and safety trial on 40 patients in the University Hospital in Basel, Switzerland, on a first not very potent version of the product, we could show 30% responder rate. In these responders, 60% more reduction in migraine days over placebo, and who's a responder could be predicted on 3 very simple biomarkers, and there was no safety concerns. But the best finding for me actually is this. Here you can see that the markers of inflammation and metabolism that define these responders are actually all improved, are all improved in the responder group. I don't know how to go back the slide, so you just have to trust me on this one. Yes, I'm sorry about this. They were all approved. Well, never mind. I'll just go on. In the recent 3 years, last couple of years, we raised roughly 2.5 million in grant money, in seed rounds, and in convertible loans, and we're currently looking for 12 to 15 million for further product and clinical development. And we have a very diverse team. For example, Dr. Tim Tranktrock, who used to be at Actelion, now Johnson & Johnson, head of supply chain and quality, an Oxford colleague of mine, a medical doctor and PhD in exogenous ketones, one of the first people in the world to actually study our key metabolite in patients, together with me and many more great people. So thanks to MigraKete, this is how I feel today. So if you want to help us fuel other migraine brains, please fuel KetoSwiss. Thank you very much for your attention.
Thank you, Elena. Wonderful presentation. Sorry about the technical difficulties, but you handled it like a champion.
Well,
So I think, is the diagnosis of metabolic migraine a standard of care today? That's the question from the audience.
no, it's not. It's a great question. It's actually our last trial where we newly defined this subgroup, and the paper will be published very soon, but it's a very new phenomenon. It's actually the first migraine subgroup that can be defined with biomarkers.
And how strong is the correlation? Is it very clear that it's a subgroup?
In our study, it's very clear, but it's only 40 patients. So we'll see how strong it holds when we do more research or larger research.
Wonderful. And a second question from the audience. They're wondering, what's the target mechanism of action and disease pathological identity? Do you know the receptors, essentially?
Yeah, so that's a great question. In a complex disease such as migraine, there are not just one pathway involved, for sure. So it's likely to be more like 10 pathways or many more even involved in even metabolic migraine. So what the compound actually does, we know there's at least 7 or 8 pathways involved where we know that they play a role, and this is based on metabolism, so providing a more efficient energy substrate for the brain. Then antioxidant, it's a potent antioxidant. This is important. Oxidative stress, and providing antioxidants. Then hyperexcitability in the brain plays a role. Inflammation is one of the most important factors, and the compound is highly anti-inflammatory. That's the most important predictive marker. And then we have other things related to metabolism, such as reducing glucose, long-term blood glucose, and so on. So there's many pathways coming together, and that's the most important thing when trying to cure a highly complex disease such as migraine, with many pathways being involved and many small polymorphisms together adding up to define the disease.
Great. And so would you define this as a biotechnology, or are you thinking this could be a supplement either?
So we're focusing on biotech, but it also could be a supplement due to the safety of the compound. We can go 2 routes to market actually and reach patients that are not as high frequent with a supplement, and for high frequencies, migraineurs offer the drug because we will not have reimbursement for all patients in a disease that is as common as migraine.
Fantastic. Thank you so much. And thank you to the audience for such wonderful questions.
Thanks a lot.